Good morning, everybody. Thank you so much for being here. It's very exciting to be here. There are a lot of participants. We have people really from all over the country. We have visitors from Canada, Germany, Australia, Brazil. So far, our first speaker is Dr. Moghekar, who's the head of the CSF Leak program at Hopkins in Baltimore. He's an associate professor in neurology, associate professor in neurosurgery, and associate professor in otorhinolaryngology. Thank you, Dr. Moghekar.
Thanks, Wouter and Connie, for getting us all together again. It's good to see everyone back with core similar interests. When I agreed to speak for this talk, I titled it CSF Myths, but I think myths is too strong a word. I think controversy is more appropriate because there are several controversies regarding CSF physiology that I will raise here. And to advance science, we need to address these controversies and not just ignore them. So let's get some facts out of the way first. So we all know in a typical adult, you have about 150 ml of CSF. You turn it over around 3 or 4 times in a day, so you make about 500 ml in a day. CSF is actively secreted by the choroid plexus, and it's passively absorbed at the arachnoid granulations into the venous sinuses. And you need this fine balance of absorption and production to keep intracranial pressures normal for normal homeostasis of the brain. And the role of CSF, there are multiple roles. There's a paracrine signaling role for CSF. Many neuropeptides, hormones are secreted at the choroid plexus. Choroid plexus also secretes transthyretin, IGF-2, BDNF, folate, vitamin C. Very relevant to this symposium, we all think of CSF as floating or buffering the brain, and it was Cushing back in the 20s and 30s who did this calculations with Archimedes' principle that your brain weighs 1,500 grams, so because of the buoyancy of the CSF, the apparent weight is just 50 grams. And when you lose this buoyancy, you feel the entire weight of the brain, and that's why you get the leaks-related symptoms. So just to recapitulate, this is a classical model. CSF is made in the lateral ventricles predominantly, flows down into the third, through the aqueduct into the fourth, then comes out of the foramen of Magendie and Luschka, goes down into the lumbar thecal sac, goes around the superoconvexities, and through arachnoid granulations gets absorbed into the venous sinuses. And this is what we've been taught, forever. Let's see if the video works. May not. That's okay. We can. So it was just a video animation showing how the CSF flows through the different. Oh, so there you go. We missed the first few seconds, but so it just shows how CSF gets out of the ventricular system, subarachnoid space, and into the superior sagittal sinus. And this theory, it's still a theory, and it's called as the bulk flow theory, which assumes this slow, smooth flow of CSF. We can skip this now. Trying. But these are all facts that are, all of what I've told you are just based on experiments that were based on assumptions that are no longer tenable. So whatever I have told you is actually controversial and not right, including what I said about production, absorption, flow. Everything is up for grabs. And we'll go through each of these now. So the part that CSF is secreted by the choroid plexus was based on one experiment done by Dandy in a dog, where he took out the choroid plexus on one side and saw that the lateral ventricle on that side got smaller compared to the intact lateral ventricle. People had a hard time recapitulating that experiment, but nobody listened to them. And people ignored the role of pulsatility in keeping the size of the ventricles. And people have historically done tracer studies to look at how CSF production is is done. And we'll go to the shortcomings of those tracer studies. And we'll go to what actually matters more is actually looking at water. We look at effects of choroid plexectomy. It's been done in humans for infantile hydrocephalus. Your brain is a highly metabolic organ. What's the end product of the Krebs cycle? It's ATP and water. So 20% of the water in your brain actually comes from the Krebs cycle in your neurons. And we ignore this ISF-CSF communication. There is no barrier between the interstitial fluid and the CSF. There are gap junctions, in fact, that allow free flow of water from the interstitial fluid to the CSF. And we'll talk about water permeability across the blood-brain barrier. So we'll address each of these issues. So the first method for measuring CSF production was by Masserman, where what they did was did a lumbar puncture, measured the pressure, took out a small amount of fluid. Obviously, the pressure goes down, and they calculated how much time it took for the pressure to come back. And there are multiple assumptions built into this measurement method. So you assume that during this process of lowering the pressure and the pressure building back again, that CSF secretion and absorption remains constant, and that's a completely wrong assumption because production and absorption is sensitive to the absolute pressure, and it ignores the effect of venous compression. So when you lower the pressure on the venous sinuses is reduced, CSF absorption actually goes up. So there are many assumptions in the Masserman method, and nobody trusts it anymore. So the more accurate method is the Pappenheimer ventriculocisternal perfusion method, where you cannulate the lateral ventricle and the cisterna magna, and you use an indicator, whether it be inulin or ions or whatever dye, like phenol sulfathalene or methylene blue you want, and you infuse it at a constant rate in the ventricles, and you measure the outflow and the concentration at which the dye appears, or the indicator molecule appears, and how it decays once you stop the infusion. And based on this, you can estimate the CSF production of choroid plexus. The biggest assumption in this is you're assuming that your dye behaves the same as water, right? And that's a huge assumption. In fact, what this method is measuring, and this is what all studies are based on, the 500 ml per day is based on the Pappenheimer method. It's based on what you can only confidently say is that inulin is cleared at 500 ml per day. You cannot say anything about water. Because what is CSF? CSF is 99% water. So if you want to study CSF, you have to study water transport. And to study water transport without the clouding assumptions of your indicator molecule, you have to use labeled water, D2O or tritiated water or deuterated water that behaves similarly as physiologic water. And what do those studies show? So these studies have been done a while ago, and they've been hiding in plain sight. Dr. Bering at the Brigham had done these studies. And remember, these studies were not done in pigs or rats or mice. This were done in normal humans. This would never fly in the IRB now, right? I mean, it took an 8-month-old normal infant, cannulated the arteries, cannulated the lateral ventricles, the cisterna magna, and the lumbar thecal sac in a normal infant. And he injected deuterated water to see how fast water appears in each of these different sites. And it was amazing. Water equilibrated very rapidly at all sites simultaneously. So if choroid plexus was a primary site of CSF production or water production in the brain, then you would have seen it appear in the ventricles first, then in the cisterna magna, then in the thecal sac, but that's not what he observed. He observed rapid equilibration with plasma levels at all three sites at the same time. Let's see if the laser works. So that's the arterial injection with steady state, and then different ventricle, cisterna, and lumbar levels equilibrate simultaneously at all different sites. So water transport is fairly rapid into the brain at all sites. What happens if you do a choroid plexectomy? So he did the same injections. Now, this wasn't a monkey. This was obviously not in a human. So he did the water injection before he took the choroid plexus out and after he took the choroid plexus out, and saw no difference in appearance of water inside the brain. So there was no net effect of removing the choroid plexus in what we would say is water secretion into the brain, not CSF, but it's again, CSF is 99% water. What about in a patient with aqueductal stenosis? You would think that if somebody has hard obstructive aqueductal stenosis and if everything is just being made in the choroid plexus, nothing should be coming in the thecal sac. And again, this was in a patient with aqueductal stenosis, human. Water appeared in the thecal sac at the same rate at what it was predicted from his earlier experiments. So I couldn't put it any better than what he did, that all of the brain water, intracellular, extracellular, is in rapid equilibrium with the water in the blood. And that water, or CSF, is continuously exchanging across all surfaces, both the ventricle surface, the subarachnoid space. And this is not to be confused with the water that does come along in the choroid plexus. So I'm not saying there's no water secreted at the choroid plexus. Yes, there's active transport at the choroid plexus. It drags in water, but the magnitude of that secretion of water is far lower than you would expect based on these experiments. And that water remains in, so the water is in equilibrium in the blood, in the CSF, in the Furcar-Robin spaces, in the ISF, Furcar-Robin spaces, CSF, and this assumption that, again, that choroid plexus is the primary site of CSF secretion is just wrong. Okay, so let's go to the... So where is this water getting into the brain from? It's getting into the brain from your capillaries, right? Your blood-brain barrier is huge, 100 billion capillaries. It's almost like every neuron has its own capillary. A neuron is no further than a capillary, 8 microns further than a capillary in the brain, because your brain is so metabolically active. So the blood-brain barrier is where your water gets into your brain. And this, again, has been done by experiments done by Paulson and even by Bering himself, where they injected tritiated water in normal volunteers. Again, will not fly now. They injected it into the carotid arteries and looked at how water appeared and how fast. And the permeability coefficient of water is 0.92, meaning 92% of water that gets into your brain gets into the interstitial fluid. So it's a huge chunk of water. I mean, the majority of the water goes in, which means, with every heartbeat, about 18 ml of plasma goes through your carotid into the cerebral circulation. And of that 18 ml, 17 ml of the water molecules, so 18 ml water molecules, 17 ml gets into the brain. So if you calculate the blood flow and look at how much water gets across the blood-brain barrier, it is 900 liters per day. So compare that to the 500 ml, you would have think comes across from the choroid plexus. So, and again, that's very high, even as compared to the kidneys, which are meant to filter water, right? I mean, it's orders of magnitude higher than the choroid plexus and 5 times as much as the kidneys. So what do you think is the water channel in the brain that's doing this? You would assume it's aquaporins, right? I mean, you hear about aquaporins left, right, center. But people have looked at it very closely, and EM studies have looked at it even more closely. And if you look at the nanogold EM work, aquaporin 4, which is again at the blood-brain barrier, is present at the astrocytic end feed. It's not at the endothelial cells. There's no aquaporin at the endothelial cells. Remember, water has to get across the endothelial cells to even reach the interstitial fluid. So aquaporins regulate or are involved in water homeostasis once water actually gets in. But that first barrier, that endothelial cell barrier, there are no aquaporins. Again, a common myth. So, and again, aquaporin knockout mice are not lethal. I mean, if they were so crucial in water transport for the brain, they would be lethal in vitro. And actually, people still don't know what is the water transporter at the blood-brain barrier. People know what's at the choroid plexus, but not at the blood-brain barrier. And it's likely it's not just one, and there are probably many solute co-transporters, like your sodium-potassium chloride co-transporter, that likely drag water along with it. GLUT1 has been shown to be carrying some water along with it. And there are some claudins, like claudin 2, that form a water pore channel that's selective for water and allow water to get into the brain. But again, this is still not fully defined. Okay. Now let's come to absorption, right? I mean, again, hopefully absorption is simpler. But there are several animals that don't have arachnoid granulations, and they have a nice functioning CSF ventricular absorption system. Even developmentally, in utero, we develop CSF in our ventricles before there's any appearance of the arachnoid granulations, weeks before there's any arachnoid granulations. And again, the experimental evidence of clearance is flawed because people use dyes. In fact, the earliest experiments to show that CSF is absorbed was done using gelatin that was injected at 40 to 50 millimeters mercury. So if you inject gelatin at that high pressure, like 40 or 50 millimeters of mercury, some of it is going to make its way through the arachnoid granulations into the superior sagittal sinuses. But that's not what happens physiologically. It's likely that arachnoid granulations serve as a safety valve. So if pressures do go up, there's a safety valve for CSF to get out. But that's not the primary site of absorption of CSF. Okay, so how fast does water get out? Again, experiments have already been done. Deuterated water injected into the ventricles is out in 5 to 10 minutes. So if you believed in the bulk theory, it would get out at the same rate at which it is produced, like 500 ml in a day. But you inject deuterated or tritiated water into the ventricles, it's out rapidly. And again, these experiments were done in 4 children, mentally challenged, would not fly with the IRB right now. They had, at least on grossly, they had no brain abnormality. But the moment you inject it, sorry, I need to go back. So the moment you inject CSF in the ventricles, it rapidly comes out into the circulation. And even in a person with obstructive hydrocephalus, you inject it in one ventricle, it still comes out, and you can in fact see the other ventricle, it goes up because it goes, the water molecules get absorbed into the systemic circulation and come back into the other ventricle across the blood-brain barrier. So, which shows that absorption and secretion of water in the brain is very, very rapid. And these are some experiments done in cats where Oreskovich went in and cannulated the aqueduct of a cat, a normal cat, and connected it to a tube. And all he saw, there was no net flow of CSF across the aqueduct. The water at the tip of the tube, just a single drop, kept pulsating back and forth with no actual accumulation of water over hours. We'll look at some phase contrast studies, and we'll look at some patient examples of aqueductal stenosis. So this is a patient of mine who had tremor, actually, essential tremor, and she got an MRI of the brain. Incidentally, she had no headaches, no gait problems or incontinence or anything, and she had this tectal mass, which is likely a benign tectal glioma, which we've followed now for 6 years with no increase in size of the glioma, no increase in size of the ventricles. So where is the water, where is the CSF going? There's a complete obstruction at the level of the aqueduct. There's no CSF getting across the aqueduct, and she's completely fine. And we did phase contrast imaging, not a great video, I think, but essentially we did not see any water flowing across, any CSF flowing across the aqueduct. We'll skip this video, but there's a better one after this. Oh, there you go. So you can see water coming out of the 4th ventricle over here and water from the lateral to the 3rd, but no, again, no CSF signal where the aqueduct is supposed to be. Another patient, again, well described in the literature, 40-year-old with aqueductal obstruction from a pineal cyst. This patient, in fact, had normal ventricles, not even big ventricles like my patient. And for 40 years, where is that CSF going? It's getting absorbed through your parenchyma into your capillaries. Okay, now, very relevant to this, right? This brain floats in CSF. So some problems with those assumptions, again, which we should address. You have about 100 ml extraventricular CSF, the rest is in your ventricles. CSF density is 1.007. Brain mass is 1,500 grams, and brain density is 1.05. Brain is still 85% water, right? So it's not surprising. So how is it physiologically possible for a heavier object to float in a less dense fluid, right? It's just not possible. The brain is actually suspended, and there are many things. With new high-resolution cyst imaging, you can see all these cool membranes in the subarachnoid space that suspend the brain. There are blood vessels that traverse the subarachnoid space that are anchored to the dura. There are cranial nerves. There are 12 cranial nerves that are at various levels, and the tentorium. So there are many different mechanisms for your brain to stay where it is without invoking CSF. And again, this is a membrane of Liliequist. Any neurosurgeons out here who do endoscopic third ventriculostomies will encounter this. It's below the floor of the third ventricle, and it anchors the brainstem to the clivus ahead. So what is the role of CSF, right? If it's not floating your brain, yes, it has paracrine function. The main function of CSF is actually that it is a pulsation absorber for the brain. And to explain that, I need to tell you a bit about some arterial physiology, actually. So remember, when your heart is pumping out blood, your systolic blood pressure is around 120, diastolic 80, blood flow is pulsatile. But your capillaries are thin vessels, single-walled thin vessels. If they were exposed to that high pressure of 120 by 80, they would burst, right? So how does the body reduce the pressure from pulse pressure of 40 and an absolute systolic pressure of 120 to a pressure of 10 millimeters and change the flow from pulsatile to continuous at the capillary level? So how is this transition achieved? And physiologists in this room will know of this Windkessel phenomenon, which is with every heartbeat, essentially your aorta and your elastic arteries expand to capture that volume and then relax slowly to allow the blood to propel forward. And this mechanism is present in all arteries that are elastic, so all your major arteries up to the level of the arterioles. So as you can see from the pressure diagram, so that's the pressure in the just in the aorta, and by the time it reaches down here at the capillaries, the pressure is down to 10. And that's because of this Windkessel effect. So the arteries expand and then slowly relax, and that propels slow blood flow forward, so your capillaries are not injured. So have you ever wondered why are the major blood vessels of the brain on the surface and not in the center? All other organs, arteries go bang in the middle, like the kidney, the liver, and then supply the organ from the inside, right? But in the brain is unique. The artery, all the major arteries, your ICA, MCA, PCAs, are all on the outside, and they supply blood inwards, right? Because the arteries expand. These are elastic arteries, right? So they expand with every heartbeat. If the arteries were from the inside of the brain, that would injure the brain parenchyma. But because these arteries expand in the closed cranial wall, if it were to happen with every heartbeat, your pressure would go through the roof, right? So what CSF, because it's not viscous, what CSF does is it displaces from the subarachnoid space into the thecal sac, because your thecal sac is a compliant structure. It's expansile. So with every heartbeat, in fact, as the arteries in the subarachnoid space expand, an equivalent volume of CSF gets displaced into the thecal sac. So the CSF is a means for equalizing pressure during the cardiac cycle. So that's its main role. It converts the pulsatile energy from the intracranial compartment and makes it and transduces it to the thecal sac. And this is just an example, and this has been looked at with phase contrast imaging now very well by Olivier Paladin. So with about 20 ml comes in with every heartbeat. Instantaneously, 2.5 ml gets down into the thecal sac. About 16 ml gets into your venous system, and that achieves equilibration of pressure with every cardiac cycle. And again, these are well-described studies. I'm going to skip for now. We are running short. So you see, so when we look at actual phase contrast imaging, you see this pulsatile flow. It's not a smooth flow. In fact, the flow goes in and out, back and forth. So flow is actually pulsatile in the aqueduct, at the foramen magnum. It's not like a smooth, continuous flow that you see in theories of bulk flow. And you can see this. This is going in and out of phase at the aqueduct, and that shows that the CSF is oscillating. It's not flowing just like in one continuous direction. And why this matters, for several reasons. We have MRI now, and instead of using an indicator molecule, you can actually use MRI and water as an endogenous contrast. And we just published this work in 2018 called as webcast MRI. I mean, the reference is down there if anybody wants to look at it. And we were able to recapitulate the radio tracer experiments using just MRI, where you pulse label the water in the arteries and look at how those water molecules then translocate across the blood-brain barrier without any contrast. So this is not using GAD. This is just using water as a contrast medium. And there are certainly applications of this in patients with CSF leaks. And I think this method could be easily applied to look at where the water is going. Another important thing is pulse encephalopathy. So remember, in patients who have CSF leaks and they don't have that CSF to translocate those pressure changes with every heartbeat, what's happening to that arterial pulse? There's no buffer anymore, so all that arterial pressure, the 40 millimeters of mercury pressure is being transmitted to the capillaries. And that's where you're getting the injury to the blood-brain barrier, to the end-stage capillaries. That's the leak. That's the cause why CSF protein is high, because the blood-brain barrier becomes leaky because of this high pulse pressure of the capillaries. And no wonder these patients have cognitive impairment, because there's no CSF now to buffer. And why do we see some patients, they have this huge ventral leak, but they're completely asymptomatic? We need to be looking at how water flux is happening in these patients. Are they compensating for it because there's more water flux into the intracranial compartment? So if we need to understand CSF, we need to get rid of all those old misconceptions and try to understand water. And I couldn't have said it any better than Bertrand Russell. So I'm glad there are no questions now, but and there is a long list of other things we can go over at the next symposium. Yeah. Thank you.